We'll go ahead and get started so we can stay on track with our agenda today. Hello, everybody. My name is Rodney Sutter, and I'm the Program Director of Therapeutic Studies at the SWOG Data Operations Center in Seattle. I'd like to welcome all of you to the first virtual SWOG Clinical Trials training course. With any luck, we'll be back on track to provide a formal training course next spring. Before we get started with our presentations, I just wanted to take a few minutes to discuss the goals of the training course, go over our agenda, and briefly cover the ORP manual and the CRA workbench. Next slide. So the goals of this training course is to introduce the fundamentals and expectations of SWOG and NCI policies and procedures to new CRAs. I have the word new highlighted in red because we do recognize that a lot of you have experience at other institutions prior to this. So this is just procedures that are specific to CRAs that are new to SWOG. We'd like to illustrate the process for protocol development, data submission, specimen submission, SAE reporting, and audits. And finally, to provide the foundation for you to efficiently perform your responsibilities as a SWOG CRA. Next slide. So here's our agenda for the training course between today and tomorrow. Normally, this program would be completed in one day in person. However, our virtual meetings are limited to a maximum of 2 hours each. So we decided to remove a few of the presentations, condense down some of the exercises for the practicum, and then we broke up the entire course into 3 sessions. Following the presentations in the session, we will take a break for an hour, and then the next session will start at 2 o'clock Central Time. The third and final session is scheduled for tomorrow at 10 a.m. Central Time. If you didn't register for all 3 sessions, we manually enrolled you in each, as all 3 need to be attended to receive a certificate of completion. There are some of you that indicated you only wanted to attend the first 2 sessions, so you were not added into session 3. In session 3 tomorrow, all participants will be assigned to smaller breakout rooms for you to engage with your facilitator and others while completing exercises specific to enrolling patients and filling out SWOG forms. Next slide. I'd like to emphasize that in addition to this training, that you'll find the ORP manual as your main resource tool to help you in your day-to-day tasks as you work on SWOG trials. ORP stands for Oncology Research Professional. It's a title unique to SWOG, representing a joint CRA and nursing program. I strongly encourage each of you to take time to read through the ORP manual and definitely share the information with others at your institution. And the manual itself can be accessed online via the CRA workbench. Next slide. So I just brought up the CRA workbench. We recognize that a number of you tend to bookmark the CTSU website, RAVE, maybe some other sites out there. I hope that you would also bookmark the CRA workbench. At some of our mentoring visits, we find that some members aren't aware that these resources are even out there. So I'm not going to go through all of the links here on the left. The first one I'll point out is what I just referred to for the ORP manual. Next slide. Another great link is Tools of the Trade. There we go. Oh, all right. So under the Tools of the Trade, you'll find things like the CTCAE criteria. There's a date counter that you can use and a BSA calculator. Any updates that you have for roster information, there's a form there to complete. A contact list that provides all contacts at the other lead protocol organizations. If you need to print out labels for your specimen shipments, there's a creatinine clearance calculator, and then also best practices for SWOG studies, among many other links that you'll find useful. Next slide. Another great link is reports. Next. Within the reports link, you'll find your expectation and IPR reports that Phyllis will talk about in a little bit today. Access to all the queries, both RAVE and pre-RAVE. Any patients that have been deemed ineligible at your site. Any SAEs for a given study. Registrations by race and sex, and also for a given date range. Any patients that you still have in follow-up. And protocols that no longer require follow-up. Next slide. There is a link for some training. Next slide. Here you'll find some training modules available to you, including some that are specific to metadata RAVE. But the one I was going to bring focus to is a link for the CRA Clinical Trials Training course, which is the same course that you're taking today. If there's others at your institution that are new but couldn't attend this meeting, let them know that there's access online, and then they can take it at their leisure. It'll also be helpful if you need a refresher yourself, or you just like to revisit some of these presentations after the meeting is over. Next slide. Here's a link for contacting us to access many useful email distribution lists. Next slide. So we have many committee-specific and one study-specific for LungMap email distribution lists noted here. Any of these will reach all the data coordinators that are assigned to those committees. So even if your data coordinator is out on a given day, there'll be backups that you'll be able to get in touch if you need something answered right away. Next slide. We have distribution lists that are specific to questions you have about your expectation report. If you need to contact CTSU, if you're having technical questions with our website. And then finally, there's a catch-all for the data management at crab.org. If your question doesn't really fit into any of the categories previously listed, that will reach the data operations supervisors in our department, as well as myself. Next slide. There's a link down here for joining the CRA mailing list. Next slide. And I'd like to point out that during this session, all the mics have been muted, but if you have a question for a speaker, please type this into the chat feature. If there's time at the end of their presentation, our moderators will pitch them to the presenter before moving on to the next speaker. If there's not enough time at the end of their presentation, we're going to consolidate any questions that you post, and we'll forward the answers out to all participants sometime after the group meeting is over. So at this time, I'd like to go ahead and introduce our first speaker, Dana Sparks, who is our Director of Operations and Protocols from the SWOG Operations Office in San Antonio. She'll be discussing clinical trials and protocol development within SWOG. Dana.
Hi, welcome to everyone. We really appreciate your interest in this course and in doing a good job for SWOG studies. Go on to the next slide, please. So I have the pleasure of talking to you about today is protocol development, and particularly protocol development within SWOG. Next slide. And next slide. So what we'll be talking about today is just a little bit about preclinical background, some of the different types of clinical trials that we do within SWOG, and in addition, other types of trials that we don't do within SWOG. Then we'll talk a little bit about protocol development and then the key protocol sections and some actions that can take place on protocol documents after they're activated. Next slide. So preclinical background basically refers to all of the activities in developing a new drug or a new concept for combinations of drugs or drugs and radiation or other medical specialties before patients are treated with that treatment on a clinical trial. So it could include studies of laboratory tests that can be done on cells that are grown in the lab. It could include testing on animals. It could include computer simulations on a drug's activity. And I think the important thing to recognize here is that there's a big funnel. And so many new drugs come into preclinical testing, but very few go into clinical trials in humans. And the preclinical background is very important because it helps the FDA to determine whether a new drug has been effectively and efficiently studied before any humans are treated with it. Next slide. So the different types of clinical trials that we do within SWOG, we have a tendency in our large cooperative groups to focus on phase 3 clinical trials or randomized phase 2 studies, but there are a lot of different types of studies as well. Next slide. Phase 1 trials are developed to determine a maximum tolerated dose of a new drug. Now, this is kind of a shorthand of saying these studies are designed to determine how to give the drug. So if you're doing a combination trial, what doses of the drug should be given in combination with each other that are most safe and most effective. For a brand new drug that's in a single agent study, it's to determine the dose of the drug that can be given safely to patients without giving too much. So there's, we want to make sure that we minimize toxicity and maximize efficacy. Phase 1 trials tend to be fairly small, so we're usually talking about tens of patients, 30, 40, 50 patients. They're usually done either at a single institution or a small number of institutions. The large cooperative group setting, the NCTN trials, are not really an effective way to do phase 1 studies for the most part. Next slide. So if in a phase 1 trial, a maximum tolerated dose can be identified and the drug can be given safely and has some anecdotal information about efficacy, then a phase 2 study can be developed. And a phase 2 study is really intended to look at harder endpoints, such as tumor response or extended patient survival. Phase 2 trials tend to be a little bit larger. So we're usually talking about upward of 30 to 50 to 100 patients, not thousands of patients, but in the range of 50 to 100. The endpoints, as I said, are usually related to tumor response, so a little bit of a shorter time frame for assessing the endpoint. And in general, we really want to look at a patient population that's fairly uniform. And so this is when we really focus in on the efficacy within certain types of cancer tumors. Next slide. If a phase 2 study is successful and meets its endpoint in terms of effective tumor response, then it's likely that a phase 3 trial will be developed. And a phase 3 trial is intended to be the gold standard of comparing the new treatment versus the standard treatment. In most phase 3 trials, there is a bar that is set that will indicate whether the new treatment is effective in comparison to the standard treatment. And this is where the NCTN groups, the larger cooperative groups that have many hundreds of institutions participating in them, can be most effective, is in doing phase 3 trials. Next slide. So next we'll move into protocol development. And this process is very complicated. So I want to stress that this is a simplistic slide, and it's very specific to how the NCTN groups work with our grants from the National Cancer Institute. Next slide. So generally within SWOG, the protocols are developed within our disease and research committees. And so we have many committees that work together to decide which research deserves to have the top priority for moving forward. The committee chair is a person who's really important in this process, in that they are a person who's at the top of their field and who has the responsibility for understanding what other research is being done in the United States, whether it's commercial research or grant-funded research, and how any new proposal that may come up within SWOG could fit into the research that's already been done, and making sure that we're answering questions that are important and relevant and are not being answered by another trial. So the committee chair is a critical person in determining whether a new concept can move forward. Any investigator within SWOG can propose an idea for a new concept, and if the committee chair approves that, then that concept can move forward for SWOG executive review. And SWOG executive review really looks at these new ideas to decide whether this particular research is appropriate to be done within SWOG. Do we have the resources to do it? Does it fit within our priorities? And is it something that our institutions and our investigators are accustomed to studying? If it moves past SWOG executive review, then we would submit the concept to the NCI review process. And in most cases, that involves an NCI designated steering committee. The NCI uses steering committees for a peer review process to ensure that the new studies that are being developed within the NCTN system have enough scientific rigor and answer a question that needs to be answered. Presuming that the NCI approves that concept, then the protocol may be written, and we have a whole process by which the protocol coordinators in the operations office assist our study chairs with developing the full protocol document. And finally, as part of the NCI review process, all of our studies are required to go through a full NCI review of the complete protocol and consent form. It's likely that the NCI at this point in their review process will point out some changes that need to be made in the protocol document, and we will make revisions to the study based off of what the NCI points out to us. And then hopefully we will receive an NCI approval and be able to use our grant funds to run the study. This process is very lengthy, and in terms of the timelines given us by the NCI, we try to move studies through the process as quickly as we can, and for the most part, smaller phase 2 studies usually will take 6 to 9 months for development, and larger phase 3 studies will normally take between 9 months and a year to get through the development process. And this also includes any contracts with pharmaceutical companies to provide drug or other support for the studies. Another part of the NCI approval process is review by the NCI Central IRB, which now all of our participating sites are required to use as they're reviewing IRB for NCTN studies. So there's a lot of information that's contained in a protocol document by the time it's gone through this full review process, and there's also a lot of detail that must be gathered and maintained in the document as part of this review process. And because of that, we try to keep our protocol documents as similar as possible in terms of where to find information. Next slide. These next two slides really go into kind of the timelines that the NCI has set out, and we do try to shoot for as much as we can for hitting those target timelines rather than the absolute deadlines. The NCI does reserve the right to withdraw their support for a study if we take too long in the protocol development process. Next slide. So each step of the process is given a certain number of days for development, and for phase 2 studies, we really shoot for the 210 calendar days in development. Next slide. And then, as I noted, for phase 3 trials, it tends to be longer, and we, as much as we can, try to keep within that 300-day timeline for protocol development. Next slide. And of note, the IRB approval is a part of this process and sometimes can delay the activation of a protocol depending on what the elements of the study are and whether they're problematic for the CIRB. Next slide. So now we'll move into talking about the content of the protocol document and touch on some key protocol sections. Next slide. So at the front of every SWOG protocol that has a randomization involved in it, you will see a schema that sort of outlines how a patient moves through participation on that protocol. So in general, it would include the registration to the trial, the randomization, what the different arms of the study the patient could be randomized to, how long follow-up continues, and when a patient goes off treatment, whether the treatment is continued until progression or if it's a defined treatment length. Section 5 will always be the eligibility criteria for a trial, so it's really critical to become familiar with Section 5 as how it relates to your patient on any particular protocol that you're working with. Next slide. Section 7 will always be the treatment plan. So this is where you'll find detailed information about how to treat the patient on a protocol, how long to treat the patient, what are the criteria for off treatment, and how long does follow-up take on this particular study. And then Section 8 will always be the dose modification section. So this alerts you to the toxicities that need to be monitored in detail for this protocol and what to do if a patient experiences side effects. Next slide. Section 9 will always be the study calendar. So you can work with the study calendar to identify the particular needs for your patient, when to schedule laboratory tests and scans for tumor assessment. Section 12 is an important section if it exists in a protocol. In most of our protocols, it doesn't. But if we have a need for a central review in real time on a protocol document, so we might ask for detailed pathology reports or detailed surgical summaries to be submitted separately from other data submitted on the protocol to trigger a real-time review of those data. Next slide. Section 14 will always be the data submission schedule. So this lays out the timeframes for submission of study data. And then we also include a static picture of the master form set so that you can use that to take a look at what kind of data need to be submitted in what time frames, and then the actual forms themselves are found in Medidata RAVE. Next slide.
Hi, Dana. I just wanted to let you know you have 5 minutes.
Thank you. Once a study is activated, there are several actions that can take place on a protocol. Next slide. And so an activation is the first time that a new study is sent out to sites, and you can start processing it through your local procedures and identifying patients to participate on it. In SWOG vocabulary, an amendment is a larger change on a study which could impact on patient risk or the risk-benefit ratio, and a revision is a smaller change that's more editorial in nature. We also can send out memoranda on studies, which provides additional information on a protocol without actually changing the document. And we may also send out temporary closures or permanent closures, which kind of makes sense. They sort of are self-explanatory. Temporary closure can be sent out as a pre-planned part of the study to allow time for assessment of data that have come in, or it may be sent out if there are unexpected problems on a protocol so that those issues can be assessed and it can be determined whether or not the study can be reactivated. Next slide. So this is what a study looks like on the SWOG website. And when you click on some of those downloadable documents, the majority of those will take you to CTSU to actually obtain the documents themselves. But there are additional links that are available on the SWOG website. So I'd encourage you to also access information on the SWOG website in addition to the CTSU website. Next slide. And this just shows additional information that's on the SWOG website, a summary of the eligibility criteria, some publication information, and links to our report of studies reports, which are put out every 6 months and summarize the data on each individual trial. Next slide. And I just wanted to mention briefly, there's some information about the NCTN Trials Network if you're interested in just being able to find out more information about how the NCTN works. Next slide. And the other NCTN groups, Alliance, ECOG, Akron, NRG, the Canadian Clinical Trials Group, and the Children's Oncology Group, which are all part of the network that we participate in together. Next slide. There's some information here on the NCI website about the NCTN structure. If you have an interest in this, it kind of helps put all of the work into context. Next slide. And then, of course, the CTSU website, where you will do much of your work regarding the NCTN networks. It's critically important to make sure that you have access to this information. This is where you'll obtain protocol documents and other documents you need to participate on a study. Next slide. So now it's your turn. If you have any questions, please let me know. I don't know, Christine, or if there's any questions in the chat box.
Not received any yet.
All right, I will walk through very quickly just so you can become familiar with some faces. Next slide. So protocol staff at the operations office include, next slide, or we did, yeah, next slide. Myself, I'm the Director of Operations in San Antonio. Next slide. We have 3 clinical trials project managers, Crystal Miwa, next slide. Andy Hita, next slide. And Gretchen Getz. And then our protocol coordinators include, next slide. Carol Laubach, next slide. Patricia O'Kane, next slide. Mariah Norman, and next slide. Laura Gildner, who both work on LungMap, next slide. Melissa Biddle is a new protocol coordinator who's working with our leukemia and myeloma committees, next slide. Katrina Morellis is also working with early therapeutics and melanoma, next slide. Chrissy Clepetto is working with our palliative care committee and the gastrointestinal committee. Next slide. Sunny Tinsley actually has just put in her resignation, but she was working with our combo match studies and with our symptom control and quality of life studies, and so we will be searching for a new replacement soon. Next slide. Chrissy Laubach is working with our lung committee and with our non-grant funded studies. Next slide. Jennifer Beeler and Taj Pereira, next slide, have just started working with the GU committee to help support GU and also our survivorship studies. Next slide. And at this point, I am finished, and I would like to introduce Leah Everhart, who is one of our clinical research data coordinators at the SWOG Statistics and Data Management Center, and she'll be talking to you about data submission, and this is really critical for those of you who are clinical research associates at sites and will be submitting data in RAVE and outside of RAVE. Thank you.
So Dana, thanks for that presentation and introducing Leah. I actually just got a message from Leah that the power went out in her house. So she's hoping it comes back on very soon. Bill, I'm wondering if we can advance past this presentation and move on to Phyllis's presentation for reports and tools to support quality data. And then hopefully if Leah gets back on, we can go back to the data submission presentation. Okay. Hopefully Phyllis is ready. Sorry about that, because I texted about it like a minute ago.
No problem. I'm waiting here, just making sure that the dog is out of the house and not bothering me during my presentation. So I'm Phyllis Goodman. I'm the coordinating statistician for Institution Performance Review at the SWOG Data Statistics and Data Management Center. I'll be presenting the reports and tools that are available to support quality data on SWOG trials. Next slide. When the grant to support SWOG activities at the SDMC are submitted to the NCI, we're asked to provide data regarding the quality of the data that are collected. Specifically, the grant asks us for data on timeliness of data submission and accuracy of the data. For timeliness, they're looking for were the required forms submitted and were they submitted on time. And for accuracy, other than charts that are identified for either auditing or monitoring, SWOG doesn't compare the data compared on the forms to what's in the source document. What we can provide is statistics on whether or not, upon review by the data coordinator at the SDMC, the submitted forms are free of queries. Next slide. So in order to facilitate data submission, in the late 1980s, SWOG developed what is known as the expectation system. This system identifies and tracks the submission requirements for study forms, specimens, patient-completed forms, and any other protocol-required items. Originally, there were only a handful of expectations that focused on study forms and follow-up, but this has expanded over the years such that now there are over 700 unique expectations definitions. So an expectation is anything that's expected to be submitted to the Stat Center and reflects the protocol-specific data and specimen requirements. These expectations can be posted conditionally, for example, based on the arm to which the patient was registered, or a stratification status, or the disease status or answers to consent questions. And this, I'm sorry, is very, that's a picture of my dog waiting expectantly outside of the coffee shop for me to come out. Next slide, please. The four categories of expectations are the initial form set, which is anything associated with the patient registration data, including those used to assess eligibility and the specimens that are due at registration. The next is post-baseline forms, then vital status updates, also known as patient follow-up, and post-baseline specimens. And expectations can be posted in two different ways: at the time the patient's registered and during treatment and follow-up. Next slide. Expectations that are posted at the time the patient is registered include the baseline forms and specimen requirements, also known as the initial form set. This includes the on-study form, the baseline abnormalities and tumor assessment forms, source documentation, including the path and op reports, baseline specimens, either for confirming disease or those for the translational medicine or banking, and other study-specific items such as quality of life questionnaires or imaging source documents. Other expectations that can be posted at the time a patient is registered are those that are due at a defined point after registration. For example, a quality of life form due at week 4 or a specimen due at week 8. Expectations that are posted at the time the patient is registered will appear on the confirmation of registration. Next slide, please. Expectations that are posted during a patient's treatment or intervention and during follow-up utilize what we call dynamic expectations. They're dynamic in that they're time period specific, and the posting of them depends on the patient's actual schedule. The due date's determined based on the data submitted on the prior forms regarding when the patient was last seen, the time period being covered, and when the next visit is due. The submission schedule of the forms is based on data stored in the SWOG database regarding the expected patient visit and assessment times as specified by the protocol. For treatment trials, typical forms that use these, these dynamic expectations include the adverse event, treatment, and follow-up tumor assessment forms. But we've recently included other studies and new cycle-specific forms that use these dynamic expectations. And some of these include on 1802, a prostate study. They have cycle-specific PSA and testosterone, and a number of lung studies use cycle-specific lab value forms. Next slide, please. Another kind of expectations that is posted is for vital status updates, also sometimes referred to as follow-up expectations. Here we're looking to see if the patient's follow-up schedule or the expected visit schedule is being adhered to. The frequency and length of follow-up is protocol specific and can be found in Section 14, as well as sometimes in Section 7. And when we calculate follow-up length, we consider both patients receiving protocol-specified treatment, as well as when it's complete and patients are continuing to be followed. Next slide, please. For posting vital status expectations, we use the patient's last contact date to calculate the patient's time since last follow-up. For determining last contact date, for studies activated prior to 2019, the last contact date is updated from any one of a number of forms that have a date on them, and this includes the treatment form, the AE adverse event form, the follow-up tumor assessment form, and the SWOG follow-up form. But because this process of gleaning the last contact date led to potential errors and additional data collection for sites, for newer studies, we've implemented the use of a single vital status update form. The vital status update form was introduced in 2018 and was first used on the studies S1802 and 1806, and please hit enter, but it is now the wave of the future and is being used on all studies going forward. If you hit enter twice now. That's a little kludgy, and enter again, please. The form is very simple and only asks for two questions: the patient's vital status, that is the patient alive or deceased, and the date of the last contact date. Enter. The most important thing about the submission of this form is that it needs to be submitted prior to any other data related to this visit. It's critical because the submission of the form sets the last data within RAVE. And if a form is submitted with the date after the last contact date, a warning will be submitted. So most important thing is this needs to be submitted first when you're going to enter data for a time point. Next slide, please. Specimens have become a larger part of SWOG protocols. They're used for multiple purposes, including eligibility, translational medicine studies, and banked for future research. Specimen types include blood products, often multiple collections using different kinds of tubes, tissue products, urine, and more recently, some stool samples. Participants can opt out of some of the specimen collections during the informed consent process. This is recorded in the SWOG database as part of the registration process, and expectations for these patients who opt out will not be posted. Specimens that are collected at baseline are part of the initial form set, and other post-baseline specimens are a separate performance criteria. Next slide, please. The expectation due dates are determined by the protocol. For newer protocols, we've been striving to adhere to the CTSU guidelines, which are currently 15-15-30. Baseline data are due within 15 days of registration, on-treatment data within 15 days of the visit or the knowledge of the event, and off-treatment data within 30 days of the visit or knowledge of event. However, you always need to check Section 14 for data submission timelines, as older studies will be different. And for specimens, including the baseline and the post-baseline, the timeframes are either posted in Section 15 or Section 14, but typically, blood products and urine are due within 15 days, except for the kinds of tubes such as Streck tubes that need to be shipped within 24 hours, and tissue is due within 30 days. But once again, always check the protocol. Next slide, please. There are a number of reports that the SWOG produces to aid in the submission of quality data. These include the Expectation and IPR, or Institution Performance Review Report, and I'll be talking about those in more detail in just a minute. But I'll also discuss just briefly the Query Report, the Ineligible Patient Report, and the SWOG Patients in Follow-up. Next slide, please. All of these slides can be found on the SWOG CRA workbench, accessible via swog.org, when Rodney talked about this a bit. After logging into the workbench, navigate to the Reports link, which is midway down on the list of links. The reports have been organized into several areas, and these reports are all under Site Management. Next slide, please. Next slide, please. On the Expectation and Institution Performance Report page, there's a news section. Enter, please. And where information pertaining to these reports is posted, anything new. Enter, please. When you navigate to the bottom of the page, you'll see the links for your site. Of note, reports are generated separately for patients registered to your site credited to SWOG and those credited to another network group. This may change in the future, but for now, there are 2 sets of reports. Next slide, please. There are also 2 versions of the expectation report, one that's generated monthly and the other that's available on demand that we call the current report. The monthly report is static and is generated at the beginning of the month. This is the report that is emailed monthly to the head CRA and PI. The current reports are dynamic and reflect the data in the database at the time a report is run. Both of the reports are downloadable to Excel and identify those expectations that are overdue enough to affect the institution performance or compliance statistics. The category to which the deficiency is counted is coded. Next slide, please. The current report is filterable with the ability to show only those items that are overdue or counting negatively in the compliance statistics. Patients are identified by their ID, initials, and information on the study, the registration date, the last contact date, and the patient status. And then on the right, we've got the due date. We have the due date, an explanation as to what the expectation we're looking for is, or the qualifier. And then the last columns, we show the number of days overdue. And if something is upcoming due, because the report doesn't just show overdue, but upcoming due, the days overdue will be blank. And then if it's overdue enough to count towards your IPR compliance statistics, the code will be right there in the right underneath that IPR column. Next slide, please. Here's how the different expectations apply, the different kinds of expectations display. For the dynamic expectations, those that are posted with reporting periods and due dates dependent on the patient's last visit, they appear as such, with information about the time period being covered. Here are a few examples. Next slide, or enter, yeah. Here is a different kind. This is the baseline expectation for the baseline tumor assessment. Enter, please. And then the follow-up or vital status expectations, just asking for an update to the patient's vital status, last contact date, and next slide, the blood. Next slide, please. And I'd just like to point out that for specimen expectations, we're trying to be as specific as possible so that when you go to the specimen tracking system, which will be discussed later, it'll be clear what we need you to do. Next slide. So when the time comes to submit a specimen, you use the Choose a Specimen page, can be very long when you hit this page, and it can be hard to find the specimen you're trying to submit to resolve that expectation. Next slide. You should go to the filter at the top for the specimen submission filter, and that will greatly reduce the number in the list and make it easier to find. Next slide. So you can see here now, once you put in relapse recurrence, it gets to those 3 specimens that we were looking for, rather than looking at that full long list. We've been working hard so that language in the expectation and what's needed in specimen tracking match as closely as possible so you can identify what is needed. Next slide. Really important, if for some reason a specimen cannot be collected, either because the patient refused, you forgot, the material wasn't available, or maybe a global pandemic, notify us using the notify that a specimen cannot be submitted function. You'll be asked for the reason, and using this will resolve the expectations. So when you get your report, you see something that's really overdue, and you realize, oh, we never collected it, this is how you resolve that expectation. Next slide, please. Institution Performance Review, or IPR, are the statistics we use to monitor and measure an institution's compliance with the data submission requirements. We look at the metrics to assess whether or not the required forms, specimens, source docs, etc. have been submitted. Policy 33 sets the compliance standards for the group. The IPR reports run monthly, and the report contains the compliance rates or percentages for each of the categories of data submission. The scale goes from 0 to 100, where you're striving for 0. 0% means there's no overdue data. 100% means everything's overdue. Next slide, please. Matching the expectation, there are 4 categories within the IPR report: the initial form set, post-baseline forms, vital status updates, and the specimen submission. And as noted before on the expectation report, overdue items contributing to the delinquency in the specific category are identified. The codes and definitions can be found on the expectation and IPR reports page. The IPR report is posted on the SWOG workbench, on the CRA workbench, and is mailed to the PI and head CRA of the LAPS, the member, or the NCORE site. Next slide, please. Compliance levels are stated in the policy, and it's on the expectation and IPR reports of the SWOG CRA workbench. So a site is out of compliance if on the initial form set more than 10% of patient registrations are more than 30 days overdue, post-baseline forms, more than 5% of forms are more than 60 days overdue, vital status updates, more than 15% of patients are more than 60 days overdue, and post-baseline specimens, more than 10% of specimens are more than 30 days overdue. So once again, not like a report card, here you're striving for 0% across the board. Next slide, please.
Hi, Phyllis, you have 5 minutes.
Thank you. Here's what the report looks like. Once again, next slide. If you're out of compliance, you will have an asterisk indicating that category. Next slide, please. The consequences of repeated delinquency can result in a suspension of an institution to new registrations. If you're out of compliance in one category or more for 1 month, nothing happens. If it's 2 months, 2 consecutive months, a warning email sent to the PI and head CRA. The email identifies the category and specific missing items, and the PI and head CRA are asked to address the delinquency. If it goes on for another month, so that it's 3 consecutive months, the email becomes sterner. You're warned there's a risk of suspension to future registrations, and you're given another month to get your measures into compliance, and if not, you may be suspended. Next slide. So I'm going to touch on a few of the other reports provided by SWOG to help with your data quality. The first is the query report, and these are queries posted by the data coordinator on the review of the patient's data. The queries are currently not tied to the expectation, missing forms, or IPR. However, it will be a future component of site performance, as clean data is just as, or maybe even more important than just submitting the form. The query report is located on the reports link of the CRA workbench and is also pushed out to the head CRA and PI monthly. Next slide, please. Here's an example of the query report, similar format to our other reports, a filter area at the top so you can focus down on the kinds of queries you're looking for, information about the RAVE, if it's a RAVE study, the folder it's in, the date of the query itself, and once again, as most of our reports, exportable to Excel. Next slide, please. Patient eligibility is important as it determines whether or not the patient will be included in the primary statistical analysis or other secondary analyses. Once again, currently ineligibility rates are not part of site performance, but could be in the future. It's really important to check your list of ineligible patients in case there was an error in these data that made your patient ineligible when in fact they weren't. These are the codes that are used to indicate different levels of eligibility. Enter, please. N is clinically ineligible, enter. So you need to check that the ineligibility was correct, that the data making the patient ineligible was correctly entered. If it wasn't, fix the error, contact the study team so they can re-review the data and update eligibility if necessary. Next. NI is insufficient information to determine eligibility. Next. For example, a required form wasn't done or within the appropriate time frame, so while there isn't information to make the patient ineligible, full eligibility cannot be confirmed. Next, NR is the patient's ineligible, but it's reversible. Next, if the missing data or form is submitted. Next slide. This is the introduction at the beginning of the patient ineligibility report. Next, the codes are here, a link to the code so you don't need to always remember. And next, this is what the report looks like. Once again, filters at the top, the reasons at the bottom, and next slide, please. Another report is patients in follow-up. It's a listing of patients. Next. It actually hit enter 3 times. Perfect. Oops. Oh well. So that report was, this report shows you patients that are being followed still, and on the far right, next, there's the follow-up stop date. Next. And this is the date that follow-up is no longer expected beyond, and so expectations will no longer be posted beyond that date. However, old unresolved expectations will continue to appear. Next, and very briefly, a summary of the reports available on the CTSU and the data quality portal. They can be found next at the Data Management tab at the top, and they provide information on delinquent forms and outstanding queries. The report includes RAVE studies, not the older pre-RAVE studies, and also includes RAVE studies from other network groups. A nice feature of these reports is you can deep link or get directly to the specific missing form or query. These reports are primarily, the data delinquency reports do not exactly duplicate what we have in our SWOG reports because our definitions for timeliness and the infrastructure behind the reports work differently. We're working on making these reports as close, the DQP and SWOG reports align more closely, but currently performance is measured using the SWOG reports, and if there's a difference, you should work towards the SWOG reports. Next, a detailed presentation regarding the differences can be found on the SWOG CRA workbench in this link. Next, I'm kind of running out of time, so just a quick thing. Do the auditors use the reports? They use them to support their work, but they are not part of the formal audit report. The NCI and other network groups, currently they are only used by SWOG, and it's not routinely given to the NCI or other groups, but could be if they asked. And lastly, this is the link for if you have questions, who to contact. And next slide, my last slide, is that high quality data are essential for our good studies. And we really appreciate all the efforts that you put in making sure that we have that high quality data. Next slide. And that's all I have, and I'm out of time. I kind of ran over. Thank you. Rodney, do you want me to introduce the next? I'm not quite sure who's next right now.
Yeah, we're going to go ahead and continue with Connie. Leah is actually, her entire block lost power. So she packed up and she's headed to her husband's office. So hopefully she can read out from there once she gets there. So let's continue with Connie for now. Connie, you might be on mute.
There, someone help me. I couldn't get it to go off. Can you hear me now, Rodney?
Yep, can hear you.
Okay, thank you. Well, it's my privilege to talk about a favorite topic of mine, which is long-term follow-up. And so our first slide is your mission, patient participant, long-term follow-up. And there's no if you choose to accept it because it's kind of our job. So in this little segment of our training course, we're going to talk about the why, who, where, how, when, all the details. Next slide, please. So many of you may be new, and you may have picked up cases or charts or patient follow-up duties from other people who aren't even any longer there. This presentation hopefully will talk a little bit about what is long-term follow-up. In our world, so much of the energy gets spent on treatment, active therapeutic follow-up, patients that are right in the deep weeds of it all. But I think sometimes we overlook the importance of the longer term too. It's all part of the same essence. As Phyllis so aptly shared in the previous segment, we need the data, and the way you tell the story for the patient is via the data. So it's really important that we fill in the blanks. This is kind of like watching a movie. If you don't see the end of the movie, you never know what happens. And long-term follow-up is like that. It's that last missing piece of the puzzle. It's the last chapter in the book. It's the last segment of the movie, the last 15 minutes. Without it, our patients' stories are incomplete, and they're counting on us to tell the story. Generally, we call long-term follow-up that phase of follow-up. It begins when protocol treatment is either discontinued or completed, when we've resolved questions about toxicities that happened while they were on treatment, we've determined response to therapy, and it may vary if there's an observational study. Next slide, please. There's always an anchor to hang your hat on. We're going to come back to this, but I want to call it out so that you notice it and think about it and remember it forever and ever and ever, amen. But there is a policy that's really important called policy number 30. This is a section that, as far as policies go, it's kind of nice. It's only a page and a half long. It's very clear. But I want you to think about policy number 30 anytime there's a change or transition in your patient's journey. And we'll talk more about the details. Next slide. It's important to remember that clinical trials are still questions. Answers are pending, and that's the purpose of the work that we're doing. And long-term follow-up helps put the pieces together. And part of what we're doing through this segment of follow-up is to assure continued medical surveillance for patients, making sure there's meaningful end results reporting, capturing accurate survival data, finding out if they're still alive, has their disease recurred, or what is the status of disease, and then especially looking for long-term adverse events and treatment-related malignancies or new malignancies. Often, not often, but sometimes there are things that we didn't know, that we couldn't predict. Life happens, and so it's an important piece of the puzzle to know all the details. Next slide, please. Generally, after active treatment is completed, follow-up is typically every 6 months for the first 2 years and then annually after 2 years. Phyllis talked about section 14, but I just want to stress it again. Section 14 of the protocol talks about data submission. Be sure to look at that. I think what we've learned over the years, and being one myself, a coordinator who for years relied heavily on the study calendar, we don't always remember to look at section 14. But section 14 is really important in that it outlines what that protocol specifically is looking for you to submit and when. You also want to notice when you're reviewing the protocol in light of your patient's journey, you want to look carefully for how long you need to follow up. In the olden days, they were all for life. Some studies, especially newer studies, may be more prescriptive in the timeline that they want you to follow up. But make sure you know how long you have to follow up, especially if you end up with all the follow-up cases on your desk. It's not one size fits all. Nothing is one size fits all. So next slide. It's important that you have a process to keep track of this. You know, when you start, you think, I know this patient really well. The first patients you take care of, you remember for the rest of your career in research, but then the cycle never stops. It keeps moving, and more patients come in your queue, and it's really great because we're offering this really important intervention, but over time, you can develop quite a mountain of data needed. So whatever works for you, but especially some key things to remember. You want to track by date of last contact, of course. You want to talk about all the reports that Phyllis shared with you. SWOG is really great at giving you tools to keep track of what's going on. CTSU and our data quality portal is another asset that we have, although, as Phyllis said, SWOG trumps for now. Keep looking to see what's needed. And then whatever internally you can do to keep track of what's needed, when, where, and how. If it's a tickler system or you have a calendar that you keep track, when I started, I had an index box with recipe cards in it. That tells you how old I am. But if it works, it works. In our program, we use a clinical trials management system, which correlates patients' journeys with what's going on with the life of a protocol. And your system may have something like that. And if you're new, maybe you don't know that you do. So just find out what options you have. Databases, spreadsheets, other things, but do absolutely set up some way for you to know what's next due. Next slide, please. When you're tracking follow-up, just some really key important areas that you might look for data. And we'll talk about patients themselves in a minute, but for sure the data itself, the record keeping that goes on, the clinical data. Some key places to look include hospital records, treating physician records, a referring physician, a family practice office. You can also call or send a letter to a patient. Generally, our patients very much like to be involved. With the advent now of things like long-term follow-up clinics, those are also good resources that you may not even know about in your system to look for, but any place that you can go to learn about what's happening with your patient. Next slide, please. As you're following this journey along, make sure you're documenting what you did, what you found, what you learned. And some of the things you're looking for would be vital status. What was the date of their last clinical assessment or their disease assessment, and what did you find? Cancer registries can be a great place to look for some of this information. Has there been progression or recurrence of their original cancer? Have there been new cancers? Are there subsequent treatments that have been added? Has there been myelodysplastic syndrome? Have there been other late effects, long-term adverse events that need to be reported? Those are all things to really keep your antenna high and look for and watch for. Next slide, please. One thing that we do refer to patients as lost, and I always struggle with this because the patient's not the one who's lost, we are, but we do call them lost to follow-up patients. And especially with COVID, this is a timely topic to bring up right now because I think we have yet to see the full impact and implications of people losing jobs and losing homes and losing insurance and being afraid to go to the clinic because of COVID. And there's just so many things happening. So just some suggestions. We're going to talk about strategies to prevent losing our patients. And again, they're not lost, we are. So preventing us losing our way and tracking how things are going for them. Next slide, please. Some of our best strategies simply boil down to communication, which is never simple. And this huddle is everybody and everything and anything that helps take care of our patient, including the patient. It might be the clinic staff, it might be the office people, it might be just different resources over time. It might be the patient's family, but become part of their journey with them. I think in research, we have one of the privileges that to me has meant the most over the years, which is that we're one of the threads that follow the patient's journey all the way through. There are very few other aspects of their clinical care course in cancer care that get to be with them, from worrying about diagnosis to being diagnosed, to making treatment decisions, to seeing how those play out, to following along for years after in some cases. I met some patients in the 1990s that I still keep in touch with to this day. They think of our office as a resource if something happens in their family. It's kind of a really cool aspect of being part of long-term follow-up for our patients, is that you get to know the end of the story with patients. Next slide, please. We're talking about prevention, preventing gaps in long-term follow-up. And it all starts by being proactive, and it starts at the beginning. Ideally, if you're just helping to enroll patients now as you're beginning your journey as a clinical research professional, you want to make sure as you're working them up and getting them registered, once you have consent, that you're capturing data related to how to find them. You need to know how to find them, and they need to know how to find you. But if you're just coming into this midstream and the patient's finished with their treatment, they're now in some active follow-up mode, the beginning is still the beginning for you. So don't lose track of where you are, and compile what you can, begin to learn what you can, assume changes will happen. And so if you can compile information in a way that it sets up maybe a worksheet or some tool that you can develop for yourself, so that over time you can pick up that chart and remember where they last left off and what they need next. One of the very simplest ways to do it is to pull the study calendar that I mentioned earlier and just simply use that and write on it that on December 17th, they were at their 6-month visit or their 3-year follow-up visit, or just ways to remember. Get to know your patients and their journey. Sometimes in this world, especially now with doing remote data entry, remote everything, some of us haven't been in our offices much at all since COVID hit, for example, but if you're following remotely, you can still use phones, you can still talk to patients, get to know them a little bit and make notes, because again, after a while, the mountain gets pretty big of how many patients, but remember their family, remember their kids, remember their mother, remember their dog, that they like to go fishing every spring, or things about them that will help you connect with them on a deeper level over time, and it builds a stronger bond. Confirm and update contact information at every visit. Verify the plan and timeline with them for next follow-ups. Our patients are very invested oftentimes in what's happening next, and I think letting them know when they're due next as well, in case you don't get to them, encourage them to call you. Build in handoffs. Set it up today as if you might not be here in 6 months, and if you are, great, you're ahead of the game, but if someone else has to pick up your chart, can they figure out what to do and what's next? Next, please, next slide. These are just some examples of the type of data you can often glean from a chart. Next slide. And ideally, you want to pull out what do you need to know in research. Next, please. This is an example of a participant information sheet you might create and put in the front of your chart, but if you complete it at the time of consent, review it every year with them, or at the time of a transition, like they tell you that they're moving or they're going to go south for the winter, or whenever they share a change, just making it simple so that you can find this stuff in one place. Next page, please. Next slide. This is kind of an old school strategy that we used to use heavily in prevention studies, but I would endorse it also in this domain for any type of study. But talking with your patients about how important they are to this work, how important the stories that they tell us are. And so asking them to share with us if there are 1, 2, or 3 people that if we can't reach them, are there others in their life that know how to get a hold of them? We've committed to share with them updates about studies as well. So if we can share with them that we need to reach them sometimes to let them know if there's been a study update, and let's say their phone number changes or their cell phone carrier goes out of business, or who knows, life will happen. But just more than one way to reach them. And most of our patients have been very willing to share this information with us. Next slide. Do keep in touch along the way. We talked about building a bond. Another way is to physically see them. Even if you're not the clinic nurse, and most of you won't be, because often those staff are busy taking care of patients in the clinic. But in reality, if they're in the clinic and you can see them in the waiting room, or maybe they're in the exam room waiting to see the physician, and you can just stick your head in and say, hey, how's it going? How's your trip to wherever coming along? Just say hi. Birthday cards, notes, little things like that go a long way toward helping our patients know that we value them, we appreciate them, and we know what their life is about. Helping make it easy for them, appointment reminders, postage page envelopes, simple for them to reach us, whether that be putting your business card in a laminated thing with a magnet that they can stick on their refrigerator, helping to save it in their iPhone. Just make it easy, really easy for them to reach you. Next slide. Foster good relationships. People that know your patient are really good resources as well. And you want to make sure that they don't forget that your patient's on a study and contributing to a very valuable body of work. So your physician's office, the personnel there, other people at the front desk, cancer registrars are a big help. Nurse navigators and other navigators can be helpful. Genetic counselors, anybody who's interfacing with your family. In spite of all that, sometimes we absolutely run into brick walls. Next slide, please. So there are some detective strategies we can use. Next slide. And there are some deeper dive looks and ways to find people in public domain if you can't find them through the traditional medical and other clinical resources you have access to, and the patient's maybe not getting back to you. There are some other ways you can look for people to, again, determine vital status and survival. That would include things like social media, registries, we've talked about that, state EMRs, family members. The next few slides, just page through those really quickly, if you would please, forward. Internet resources here, and again. These are included in your packet. One more. These are all opportunities where you can search in public domain to see if there's information out there about how your patient is doing. Are they still alive? Has there been a death? So again, when there's a change in status, let's stop and talk for a second now about policy number 30. The responsibility for patient follow-up. You've heard from Rodney and you've seen from Phyllis a little bit about how to find some of these things, but if you log into the SWOG member site, swog.org, look for policies and manuals and find policy 30. I put the word all in red in 2 places. If your institution's involved in SWOG, whether it's a staff member or the institution itself, all institutional and individual participants in SWOG are responsible for the follow-up of all patients registered by the institution and or the individual at the institution, as long as the patient remains alive or for a protocol specified length of time. The commitment to patient follow-up remains regardless of the funding status or membership status within the group. And we're seeing some of those changes now, whereas there are hospital mergers, there are system mergers, there are things you can't control. There are staff changes, there are realignment of affiliation status. It doesn't really matter in a way, although it makes life much harder when those things happen. But in other words, this is important. And once a patient's on study, we committed to them that we would follow them until the study was over. Next slide, please. Policy number 30 includes a few bullet points under there about multiple different types of scenarios. We're going to go through these one by one. Next slide, please. The most common one may be a patient transfer, where the patient lets you know, I need to move to Florida, or I need to move to Arizona or California or some other state. I'm in Michigan, that's why I'm using those states. Ours all move to warmer, sunnier spots, usually, than Michigan. But if they're going to go to another institution, you may be called upon to help them identify a new location. It's important to note that just because the patient moves there and we think it's okay, the accepting site also has to approve the transfer. So I want to just put a caveat on almost all of these changes. Be sure you work with your program leadership. There are implications in moving a patient from site A to site B that are really important, that have to do with IRB oversight, with communication, with follow-up and commitment. Next slide, please. The transferring institution is responsible to contact the new site. There's an online form that you have to start. It's imperative that you resolve all the expectations and queries. So my suggestion is that you also work with your SWOG data coordinator to make sure that your house is in order and things are tied off with a beautiful bow before you hit go to move the patient over. It's also been recommended that we provide accepting institutions with copies of the research record and case report forms. This used to be a little simpler before HIPAA and before lots of paper became electronic, but just make sure that within your institutional bounds that you're sending what you can. Again, it's for the best of the patient. And sometimes I know that patients themselves carry information from point A to point B. Next slide, please. If you happen to be the accepting institution, you're the one that gets the phone call from people like me in Michigan to say, hey, we need to move our patient to your site. Sometimes everybody means well, but in reality, the site may not be fully approved yet to conduct that specific study. So it's imperative that the accepting site also check the policy, that they look at the form, the transfer form with their investigator, that they make sure they have IRB approvals and other regulatory approvals, investigator approvals in place before everybody's good to go. It's also important that the patient signs a new consent form, but especially a new HIPAA authorization at the accepting institution. In the olden days, many sites included their HIPAA in their consent forms. They weren't separate, but in reality, HIPAA is a local approval. So it's important that as the patient moves to a new authorization zone, if you will, a new system, a new hospital, a new place, that there be a current HIPAA authorization in place so that no one gets in trouble for sharing information when they shouldn't. Next slide, please. This next topic is a little bit sticky, and I want to really stress how important this is, that we discuss this consent withdrawal idea with our patients in depth. Sometimes patients may say, I'm tired. I'm tired of this treatment. I'm tired of filling out these forms. I don't want to come in for any more visits. I just need a break from this, or this isn't at all what I thought. I wanted to be on a different arm of the trial. I didn't get the treatment regimen I wanted, et cetera, et cetera. But definitions are key when we talk about consent withdrawal. When we originally met with a patient to explain the study, the risks and benefits and all of those things, we pledged that we would keep them in the loop and we would keep them informed about the study that they've agreed to sign up for. What I would strongly recommend is that whenever possible, keep the door open. One of these things does not mean all of these things. And withdrawing consent, patient may say, I want to withdraw. That doesn't necessarily mean that they need to come totally off of the trial. They may not even want to come totally off the trial. So take time to explore the words. Make sure that they understand they can still be followed and that you'd like to still share information with them. Next slide, please.
Hi, Connie, you have 5 minutes.
Okay, thank you. Before you move a patient fully to the status of consent withdrawal, before finalizing this status, it's imperative that you read and review and re-review the policy. Make sure you know what it means and inform and discuss with your program leadership. I have a patient who's indicating that they would like to withdraw consent. This is what they've told me. This is what I've learned. And know and understand the implications of using that designation. For example, they may have specimens on file. They may have agreed to be contacted for future research. You don't inadvertently want to disable things that have already been done and contributions these patients have already made. And it may just be simply that they need a break. They might need a timeout, and they may want to be willing to come back at a future date to continue to contribute data. I would also encourage you strongly to inform SWOG and work with your study coordinator to verify what form to use when you're talking about this with SWOG headquarters. RAVE might need something turned on electronically in the patient file versus a non-RAVE study might be a paper, old-fashioned paper-like form, case report form that's located on the workbench. But whatever you do, talk this over with your leadership. You, the coordinator, should not independently, in my opinion, make this decision and withdraw a patient fully without going into the deep weeds with your leadership. Next slide, please. The other category that's defined in the policy, I just encourage you to take a hard look at, is loss to follow-up. And what does that mean? And what does it require before you can declare your patient lost to follow-up? I hate to say that over time there have been instances where, in desperation to clean up a QA report because people were getting beat up on audits, or they felt like their leadership was upset that they couldn't get their data metrics in good enough stead, somebody might try to find a patient, and if they couldn't, they just said, they're lost to me, I can't find them. But in reality, you can't call someone lost to follow-up until it's been at least 2 years since their last contact. There have to be at least 3 documented efforts to reach the patient. And again, my strongest encouragement is to re-review the policy, talk to your program leadership, connect with your SWOG study coordinator, and see if there's anything else you can do. Next slide, please. This is the form as it looked on the CRA workbench under tools of the trade, and you've been shown where that is. Next slide. I'm not going to go into detail on these next 2 slides, but they're in your slide packet for your reference. But SWOG is one of the few research bases who I can shamelessly applaud and celebrate and just say yay for you, because they have done some really masterful work to make life easier for coordinators and IRBs and just the whole enterprise at large. But sometimes there are times when studies are not ready to be finally analyzed. The final analysis is not done. We want to leave the door open to still collect data, but we don't need to be doing ongoing, continuous reviews on studies that are close to registration and no one's receiving protocol treatment. So take a look at this. This is a godsend. There's a list of studies that fall under this S9808 on the CRA workbench under reports and study management. Next slide. And there's also a list of no follow-up required studies. There's a lot of interest in institutions to purge files of data and information. And so you want to make sure you know how long you need to keep your records. And some of that information can be found under this tab, which is in that same section. Next slide. And Phyllis, I think, showed you this in her presentation. Next slide. To sum up our work, our patients have entrusted us with being part of their journey. Every patient counts. Telling the story is just actually, in my opinion, the joy of being an oncology research professional. Without us doing the data, without us capturing the essence of what's going on with that patient, it doesn't take very much for the study to be in jeopardy and for the details to lose their way. And I tell you what, when you're in the slog and the drudge and the grunge of doing data upon just hard stuff, this is where you can find your joy, to realize that you are changing the enterprise that we work in. You are changing the future for patients to come, because we're answering tough research questions by just strictly being disciplined, diligent, and helping get that patient's contribution through. Next slide. If we were in San Francisco in the spring like we used to be, I would say if you have questions, let's just hike across the street from the hotel to the Ferry Building and hang out at the Cowgirl Creamery and have a bowl of tomato soup and a grilled cheese. But that being said, we're not there, but I'm happy to answer any questions if anyone has any at this point in time. And thank you for your investment in this long-term commitment that we make to our patients.
Great. Thank you so much, Connie. We really appreciate your input and your presentation. We don't have time for questions after your slides, but it looks like we do have Leah back with us to present the data submission presentation. So Bill is going back to the start of her slides so she can get started.
I just want to make sure you can hear me as well.
Can hear you and we can see you.
Yay. Thanks for bearing with me there. My little technical difficulty. I'm in a new setting now with power and internet. Thank you, Rodney. So I'm going to be presenting on data submission in our 2 systems, which are the Pre-RAVE and RAVE systems. Next slide, please. As I mentioned, we have 2 data submission systems. The CRA workbench, which is the Pre-RAVE system, and it's used for all studies that were activated prior to April 2012. which is 19 SWOG studies. It's accessed, and also known as the chart manager. And then RAVE is used for all studies activated after April 2012 and is being used for 73 and counting studies in SWOG. Next slide, please. So first I'm going to talk about the pre-RAVE system, the CRA workbench. Next slide. And in the interest of time, we're only going to cover how to access the CRA workbench, how to look up patient charts, and we'll take a look at how the forms are organized. But if you have any further questions about the CRA workbench, please reach out to your data coordinator. Next slide. So to access the CRA workbench, you're going to go to swog.org and log on using your CTEP ID and password. And from the member resources link that's highlighted in the right-hand side of the page there, you'll click on the CRA workbench link. Next slide. And from the CRA workbench homepage, you'll click on the Pre-RAVE data submission link, which is highlighted on the left-hand side of this page, which is where you'll be able to search for patients. Next slide. And from here, you can look up patients one at a time, which is highlighted at the top right-hand side of the page, or you can generate a list of patients by searching the 3 categories highlighted above in the middle there. And then you can also apply the filters at the bottom to further narrow down your results. Next slide. In this case, we've looked up all patients on a particular study, and from here you can click on the patient ID number, the hyperlink there, to select a specific patient and view their chart. You can see that it's also arranged by patient number. Next slide. And the default tab that'll appear once you've clicked into that patient link is the patient information. And this tab displays the patient's registration information, vital status, and last contact date. You'll also notice that this page will show a notification if you have any outstanding queries that need to be addressed. And at the top, you'll see there are a total of 4 tabs for patient information, the default tab, but also forms, expectations, and queries. The forms tab takes you to the forms used for data entry. Expectations tab will show any outstanding or upcoming expectations for the patient. And the queries tab shows you a list of open queries for the patient. Next slide, please. So that's it for our pre-RAVE system. But like I said, if you have any questions about that, please reach out to your data coordinator and they'll be able to address any of your questions or concerns. So we'll move on to RAVE. Next slide, please. And this is just a little overview of what we'll be going over for RAVE. It'll be a little bit more in depth, but we'll try to keep it succinct. Next slide. When a site is approved for registration to a study in the CTSU regulatory system, people in the SWOG roster with the role of RAVE CRA will be sent an invitation to join that study. If you're not getting invitations, you should contact your head CRA to make sure that you're set up as a RAVE CRA. And be aware that some studies require a delegation of task logs, also known as the DTL for short. And this is completed in CTSU and identifies the CRAs and investigators that will be working on the study. And just be aware that only individuals that are identified on that delegation of task logs can enter and edit data on the CRFs in RAVE. And it also needs to be signed off annually. So if you're unable to access or edit forms in RAVE, contact your regulatory team to make sure that your DTL is up to date and that you appear as a CRA on the DTL. Next slide, please. So this is the iMedidata portal to all of the RAVE studies you have access to once you're logged in. And it'll include both SWOG and non-SWOG studies. And you'll see on the right-hand side the tasks section, which is where you'll find any invitations to join studies, and also the e-learning section below that, which highlights any outstanding e-learning requirements, which might be necessary to access forms and enter data. So you might need to complete those before you're able to log into the study. And then in the middle is the list of studies that you have access to. Next slide, please. And once you're logged in, RAVE is organized by study, which means information can only be viewed for one study at a time. And once a study is selected from the home page, the tabs across the top of the page increase in specificity from left to right in a nesting doll fashion. So it starts with the selected study, then their site, participant, and then the folders in that participant's chart, and then the forms within that folder. Next slide, please. And this is what it looks like once you've selected a study. For this example, I've selected S1826 and a test site, and highlighted in the upper left-hand corner is the list of subjects registered to this study at the selected site. And highlighted on the right-hand side is a task summary list, which displays any outstanding tasks that can be viewed by clicking the arrow next to each category. And then you'll see highlighted in the bottom left-hand side is our icon key. RAVE is very icon heavy, so that icon key appears on every page so that you can refer to that as you need to determine what all the icons mean. Next slide, please. So this is the main participant page once they've been selected. Folders containing eCRFs are highlighted in the navigation pane on the left-hand side, and highlighted in the middle are folders and their approximate due dates. But be aware that even though due dates are shown in RAVE, your expectation report and the protocol are always the best resources for determining when forms are due. And then we have another task summary list that's highlighted on the right-hand side again. But now it's specific to this patient and will only show outstanding forms for this whatever patient you've selected. And then at the bottom, I have highlighted the Add Event drop-down box. And this is a tool to add certain forms that won't generate otherwise. Next slide, please. So the forms you see in RAVE are going to look a little bit different from the eCRF or the paper CRF versions. And also keep in mind that we can create conditional settings in RAVE to only show specific folders, forms, or fields. And then depending on how you answer certain questions, they'll appear at that point. Next slide, please. So the baseline tumor assessment and follow-up tumor assessment forms are examples of these conditional forms I just mentioned. And highlighted at the top, you can see we've answered yes to the questions, does the patient have measurable lesions, and does the patient have non-measurable disease question. And this is going to generate the target lesion and non-target disease forms in the same folder once the form is saved. Next slide, please. So you can see since we answered yes to both of those questions, highlighted on the bottom left-hand corner are the new baseline forms that generated that will allow us to capture that information about the target and non-target lesions. And it also generated the follow-up tumor folder highlighted just above those blank forms. That will also contain target and non-target disease forms, which are highlighted on the right. The follow-up tumor assessment forms have also been set up to pre-populate the lesions based on the information that was entered on those baseline tumor forms. So keep that in mind, that anytime you need to change anything on a follow-up tumor assessment in terms of the lesions that are indicated, you need to actually change them on the baseline forms. Next slide, please. And in case you have questions about when or where to find specific forms in RAVE, we create a RAVE form display document for every study. And this is available within the master form set on the study page at ctsu.org. We just want to draw special attention to the form highlighted at the bottom, the off treatment notice. So when a patient comes off treatment, you need to trigger this form to populate by amending on the last cycle's treatment form, the question, will the patient continue to receive protocol therapy? If you answer that no, this off treatment form will generate and can be completed. Next slide, please. So this is an example of a typical RAVE form. The field labels are left justified and field entry is right justified. And you can notice that some fields have the required units specified, highlighted in that purple box. So make sure you're entering accordingly. And next to field entry are the status icons and edit icons. And you can open individual fields for editing by clicking the edit icon, which is that little pencil next to the specific field, or you can open the entire form for editing by clicking the edit icon, the little pencil at the very top of the form. Next slide, please. So a question mark next to a field label indicates help text. And when you click that, it'll display some data entry guidance and or clarification about that field. Most forms have a comments field highlighted on the right side of the form here, where you can explain unknown or clarify any circumstances. And finally, the save and cancel buttons are at the bottom right-hand side of the form. Next slide, please. So you'll see this message every time a form is saved, instructing you to scroll down to the form and make sure that these green check mark icons appear for every field on the form. And just remember that some forms have those conditional fields that might create new fields to fill out. So make sure you have a full set of green check marks all the way down the form before you move on. Next slide, please. And one moment, my battery is about to die. I will be right back. Okay. Crisis averted. All right. So now we're going to discuss forms with logline fields, specifically the baseline tumor assessment form in RAVE in this case. And you can see there's only one logline to start, and you can add more as needed by clicking the add a new logline hyperlink, which is highlighted. And make sure to save the form after you complete each logline. Next slide, please. And if you want to get rid of a logline, it can be inactivated by clicking inactivate, highlighted at the top of the slide there. And then you select which logline to inactivate from that drop-down box in the middle of the slide, and then clicking inactivate over to the right. And this does not actually delete the line from the table, but it strikes out the data on the logline. And It's important to notice that all active log lines that have been correctly completed will have that green check mark at the end of the log line, and the inactivated log line has an inactive icon. And similarly to inactivating a log line, you can also reactivate them by clicking the reactivate link and selecting the log line again from the drop-down box. Next slide, please. So RAVE has a special field for the submission of electronic documents, and this allows you to upload documents by searching for the file on your local computer by selecting the Choose File button and then upload the document directly into RAVE. But be aware that when uploading forms, the type of procedure selected might determine whether an associated expectation should be resolved. So be sure to choose the most specific type applicable and pay attention to the form instructions because they may instruct you on how to label specific documents in order to resolve those expectations. And you'll notice that similarly to the form on the last slide, you can add and inactivate log lines on this page as well. But you will need to, if you inactivate a log line that has a file uploaded, you do need to remove the file before you inactivate the log line. Next slide, please. So before you upload any source documents, you'll need to make sure they're fully redacted and have the SWOG participant ID number, initials, and study ID number added to at least the first page of the document. You will not be able to edit the documents once they're uploaded in RAVE, so make sure you have that all completed beforehand. And PDF files are highly preferred, and there should be no special characters in the file name, as this makes them more difficult to open. And multiple documents shouldn't be combined in a single file, and each document should be uploaded to its own log line. That might also determine whether or not specific expectations are resolved. Next slide, please. So once your upload is complete, a link to the document appears along with the green confirmation check marks. If you need to inactivate a log line, be sure to, oh, we already went over that. Be sure to remove it from the log line before it's inactivated. But also be sure to just read the instructions at the top of the form, even if you've uploaded documents to other studies, RAVE forms, because it might vary by study. Next slide, please. And when entering data onto a log line, it's important to use your mouse to select an option if there's a drop-down box to avoid any non-conformant data errors from populating, which we have highlighted there. You can't use your keyboard to complete the field like you would a free text field. That's how those non-conformant data icons will populate. So just be sure you're selecting from the drop-down box. Next slide, please. And as mentioned earlier, RAVE can be programmed to only show certain fields and forms depending on what data is entered. So let's take a look at that. Next slide. So in this example, depending on the answer selected from the drop-down box, additional follow-up questions will generate below this question after the form is saved. In this case, we've selected less than 1 year for this question. Next slide. And once the form is saved, the follow-up question that generates under question 2 is with a new field to complete. You'll notice that the status icons are not green check marks for the new field, which means that it's not yet complete. So be sure to save the form once you've completed that new field and verify that the green check marks appear for that answer as well. Next slide. So we're going to show an example of a sticky note, which you have highlighted in the bottom left-hand side. And these are mostly reminders. You just need to check the acknowledge box and save the form to make those disappear from the form. And they will show up on the task summary list until they're acknowledged, so just be aware of that. And as you can see, the presence of the sticky note does not affect the green check marks that appear on the form. So the data has been entered correctly, but make sure you have entered the data correctly, and if it requests any additional documentation, that's completed before you acknowledge the sticky note. Next slide, please.
Hi, Leah, you have 5 minutes.
Okay, thank you. Okay, so now we're going to discuss queries. A system query is automatically generated by RAVE for edit check violations and nonconformant data, which is data that does not fit the field type, such as letters that have been typed into a number field. And on the left-hand side of the screen is an example of a system query that fired because the height field was left blank. You can tell this is a system query because under the query it says open to cite from system. Another example of a system query is the nonconformant error that fired in the bottom right-hand side of the form because the date entered is invalid. And you also can see that nonconformant error icon as well next to the date. Next slide, please. So here I entered a value for the height. Always remember to check the units, which you can see grayed out the response box, which means I no longer have the option to respond to the query. And once the form is saved, that query will disappear. For any data change, you will be prompted for the reason you're changing the data. The options are entry error, new information, and per query. So just choose whichever seems appropriate. This isn't really critical. Next slide, please. You'll notice the bottom of every RAVE form, there is an option to save without submitting with the little checkbox. And checking that box will trigger a system query, preventing the data from being transferred to SWOG. And this will allow you to save the form, but not submit any information. And unlike when you choose to save without submitting on the CRA workbench, edit checks do still fire in RAVE. So even though you save it with that checkbox, those edit checks will still fire to if you've left any blank data or anything non-conformant. But DCs are trained not to look at these forms when they are saved without submitting. Next slide. And this is the query you'll get when the form is saved with that checkbox, with that box checked. And once you're ready to submit the data, simply uncheck the box and resave the form, and it will be submitted to our database. Next slide. So as you can see, all the queries have been resolved on this form, and each line has a green check mark. And you can also see there's a little arrow pointing to a yellow triangle, which indicates the data has been amended for that item. And you can click the status icon for the field, for each field, to see the audit trail if you wish. And please note that all system queries must be resolved before the data can be imported to SWOG. So it's important to address those as quickly as you can. And any expectations tied to those forms are resolved the next business day, which is something good to be aware of. Next slide, please. So the other type of query you'll see is a manual query from a data coordinator. The main difference between system and manual queries is that even if you change the data, you must also leave a response if the response box is present. You can tell this is a manual query because it says to cite from DM instead of to cite from system. There's a response box, so you can enter a response and, if appropriate, fix the data. However, because this is a manual query, a response is required even when the field is amended. Next slide, please. So here you'll see I updated the data. And you'll notice that even though I changed the data, response box remains editable. And that's your reminder that you do have to type in a response. And I could have also typed in a response without changing the data in this case. But be aware that any data entered in the response box is not saved the database, and any information pertinent to the query response and to the patient chart as a whole should be noted in the comments section or the appropriate field in the form. And queries should not be closed until they've been addressed. And if you need any clarification on what is being asked in a query, the best thing to do is reach out to your data coordinator and avoid asking more questions in response to a query. Next slide, please. So once that query is resolved, you'll see we have green check marks next to each field on the form. And the resolved manual queries will still show up on the form, unlike the system queries, which won't. So just be aware of that as well. Next slide. And you can find open queries, both manual and system queries, under the task summary list on the right-hand side, and then at the site level on the left-hand side of the screen. And then it also shows which forms on that right-hand side have those queries appearing. Next slide. And the second place you can find queries that are outstanding is on the queries report from the CRA workbench, and only manual queries can be found here. System queries won't show up, so just be aware of that. And highlighted above here, we have an example of what you can see exactly which RAVE field the query is on. But just be aware that you'll have to access the query in RAVE. You can't access it from this report. Next slide, please. So here's a list of RAVE resources available to you from the CRA workbench. You can still continue to use the CRA workbench as your portal for all things related to SWOG study patient management. You just can't submit or amend any RAVE data. Next slide. And your front line for technical support with RAVE is the CTSU help desk. Anything not related to the data you're entering or any queries you might need clarification on, which you should contact your data coordinator for that. But anything technical can be directed to the CTSU help desk. And their contact info is here and is also available through a teeny tiny customer support link that you'll see at the bottom of all pages in RAVE. Next slide. And I would say we have time for questions, but I don't think we do. But you can always reach out to your data coordinator at this phone number or that list of study or site committee emails. Thank you.
Yeah, there hasn't been any questions in chat, and we're actually out of time to take questions now. Bill's going to go ahead and pull up our final speaker for this session. It's going to be Mike LeBlanc, the director of the Statistics and Data Management Center here in Seattle. He's going to talk to you guys about the scientific impact of the CRA. And Mike, I heard that you could go maybe a couple minutes over, but it's possible that it might just time out. So
it just might time out. Can you hear me? Can everybody hear me?
Yes.
Don't worry. I'm sure in slides in one form or another will be available either by recording. So if I get timed out, that'll be what happens. We'll find out. Thanks, Rodney. Any other introductions you want to make? Good.
Nope, we're all good. For all the participants, you get an hour break after this, and then we'll rejoin for the next set of presentations.
Right, that's important to take care of that in case we end in the middle here. My name is Michael LeBlanc. I'm the SWOG group statistician. Connie showed you all the many critical aspects for successful follow-up, and I'll hope to show a little bit of the impact of doing that in terms of our trials as well as other data submission. So in this presentation, I'll describe some of the ways you are critical to the mission of SWOG and to the National Clinical Trials Network. I'm a statistician, but no matter what we do, it impacts the data you submit is critical. So here's an example of what the impact of the study data. This was a study that was completed several years ago, SWOG study S0777, a myeloma study that randomized, on the left-hand side, randomized 2 treatments, a 3-drug combination versus 2-drug combination. And on the right-hand side, you see survival curves or progression-free survival curves. The blue line above the red line, the blue line corresponding to progression-free survival for the triple drug versus the red line for the 2-drug. And if you were to read these curves, which I think is an important thing to do to understand what's going on, basically, if you saw where, excuse me, could we go back and slide, keep it as a slide, that the 30 in the experimental arm, there was 43 months median progression-free survival versus 30. Next slide. And so these results are strong because of the quality data, and not only because of a strong paper that resulted in the change of how people are treated, but regulatory impact as well. And I think the reason why I wanted to bring this up, it was because after significant additional work by the CRAs and the Statistics and Data Management Center to validate all of these results, we ended up with essentially the same results we had to begin with, which sounds like a effort misplaced, but it's not. It shows you that the high quality data that we get from you all is critical to our mission. Next slide. So I'm going to briefly remind us, clinical trials, we're trying to learn at each stage of the game. So we don't do that many phase 1 trials in SWOG. Those are ones that look at safety, early side effects, early activity. You might do many of those more as your single institutional studies. But we do a lot of phase 2 and phase 3 studies. Phase 3 studies, we're looking for more side effect assessment, refinement of doses, finding disease subgroups where we think there's the most promise. Some examples of these types of studies that you'll be putting patients on are single-arm studies, 2-arm randomized studies, other sorts of studies in that group. And then finally, the phase 3 study, that's where we do the formal comparison of new treatment to standard. And when you're putting patients on those studies, the ultimate conclusion of that study may be that this new treatment is better or not relative to what standard of care is. So that's really sort of the pinnacle of research with respect to advancing new therapies forward. Next slide. At each part of those evaluations, different types of endpoints and different types of data we collect that you're responsible for to help us sort out what's going on. There's early stage studies, biologic activity, and of course, safety and toxicity. But for phase 2 and phase 3 studies especially, there's clinical response assessments, sometimes patient-reported outcomes. Importantly, we'll show some examples later, disease recurrence and progression, of course, survival data, and the other long-term adverse events data that are collected as part of those studies. Next slide. So I'm going to talk about a couple of statistical concepts which are sort of behind all your work, things that we try to control to get good results. And those two concepts are variability and bias. And I'm going to talk about how they arise, what problems do they cause, importantly, how they can be prevented or reduced. Because typically it's not the statistical or data scientist that takes care of these in the end. It's you that really control and prevent these, because there's usually no statistical analysis to get rid of them once they're there. Next slide. So this is a plot taken from a colleague that shows these concepts in graphical form. I think several of you have seen this slide before. So on the left-hand side, accurate and precise, that's represented here by a bullseye, and all those arrows are going to the middle, basically. Accurate and imprecise, that would be high variability but low bias. Those areas are basically going to the right spot, but they're sort of spread out. Accurate, inaccurate, and precise, all the arrows are going to the spot, but it's wrong. It's going to the wrong spot. And then, of course, the inaccurate and imprecise, the arrows are not hitting the bullseye at all. So those are concepts of accuracy and precision, or related to bias and variance. Next slide. So how do we control variability? We do it by eligibility criteria. So think of this example. If results of studies which only allow patients with local disease and performance status, say 0 or 1, will be less variable than studies allowing any disease and any performance status. So in other words, we have eligibility criteria, obviously for patient safety, but it's also to make the conclusions to decrease variability with respect to our results. So if we keep those patients as similar as possible, we allow some variability control. Next slide. What's another way as statistician we control variability? Well, it's a larger sample size. So that's why one of the reasons we'll see the phase 3 studies are so large is because we're trying to reduce the variability by actually collecting more patients, having more patients registered on each arm of the study. Next slide. So your role in reducing variability is verification of eligibility, avoiding deviations from protocol treatment plans, and critical submission of complete and timely data. That controls the variability and strengthens our conclusions at the end of the study. Next slide. So what's another statistical concept? Well, that's bias. And in words, it is the tendency for a statistical result to differ on average from the true state of affairs, often due to flaws in design or conduct of a study. It's a lot of words, but basically it means no matter how much data we collect, we're going to get the wrong answer. So an example of that, if a study of treatment intended for patients with local disease includes patients with more advanced disease, the treatment's efficacy might be underestimated. So in other words, if you include the patients incorrectly with the wrong stage of disease, that estimate, no matter how many patients you put on that study, it will tend to give you the wrong estimate of treatment efficacy overall. Next slide. So solution, a strong adherence to eligibility criteria. So that is a critical aspect of bias reduction. Next slide. So an example for this can happen not just in terms of eligibility. If patients in adjuvant therapy arm of a comparative study are followed more closely than an observation arm, the benefit of adjuvant therapy may be underestimated. So what happens here is if in fact if you're following patients differently, and we'll see some examples of this hopefully in a couple minutes, that may actually bias your results and give you the wrong answer with respect to treatment efficacy. So it's critical that one follows patients per protocol on both arms of the study. That is absolutely critical. Next slide. So I show a simple example, and I'm going to take a couple minutes. Even if we don't get to all the slides of this talk, I think this is an important illustration of what the impact can be. So this is a patient registered on March the 1st, had an assessment date on May the 1st, another assessment date on July the 1st, and was seen to be progressive on October the 1st. Enter. Just a little bit of animation here. Patient B registered March the 2nd, assessment day May the 4th, assessment date again, July the 1st, and the purple arrow corresponds to a progression date on July the 1st. Enter. So the result is on patient on treatment 1 had a progression date of October the 1st. Patient on treatment B had a progression date of July the 1st. So patient B progressed before patient A on treatment 2 rather than treatment 1. Next slide. So again, let's assume we had the same patient registered as before. March the 1st registration, assessment date May the 1st, assessment date July the 1st, progression date October the 1st. But instead on patient B, that patient was registered on March the 1st, but lost a follow-up on April the 1st. So we didn't see those two assessment dates on May the 1st and on July the 1st. So that patient was lost then. So enter. That results in a progression for patient A on October the 1st, but we didn't see a progression on patient B. So that could bias the results. That could make you think, in fact, that patient B was doing better than patient A, but it was all due to loss of follow-up. At least it includes variability in the study. So this is a dramatic impact of what patient loss to follow-up can mean in terms of our conclusions. Next slide. So bias Solution is to ensure adherence to protocol requirements for follow-up examinations. And Connie gave wonderful suggestions on how to do that as cleanly as possible. Schedule, have patients return the evaluation according to protocol. Schedule and have all tests performed per evaluation. Some of those assessment dates may have been scans. If they weren't done at the right time, that could have introduced other biases as well. Next slide. So what's your role? Verification of eligibility and adding to that adherence to protocol follow-up requirements, among many other things. Next slide. So one of the things we measure is much, and you saw from the first study, is survival data or time until progression. These time until event data are very difficult to measure unless we have good follow-up. And incomplete data increases both the variability and, as I said, the potentially biases through survival studies. So it's absolutely fundamental aspect of clinical trial management is we need to get appropriate follow-up data on patients. Next slide. And I'm going to show this by one of the things we can do as statisticians, we can create pretend data. And I want to show two slides here that shows the impact of follow-up issues in respect to study. And this was a hypothetical study design, accrual over 3 years and 1 year of follow-up. And those are the two survival curves like we saw before. The yellow line corresponds to the experimental treatment. The blue line corresponds to standard treatment. And again, how to read these curves, and I think everyone should know how to read these curves because these are the results of the data that you spend so much time collecting. And if you look at these two curves at 1 year, that the horizontal axis at 1 year, about 50% of the people are surviving on both arms. Those curves are on top of each other. So the conclusion here is new treatment does not help with survival outcome. This is a result of this study that, in fact, that you should not move this new treatment forward. It's not being helpful with respect to this. Those curves are on top of each other. So so next slide. So what would happen if we lost some of those patients to follow-up on one arm? So now we're going to assume that on the experimental arm, some of those patients we lost to follow-up, but by chance, let's assume that some of those patients we lost to follow-up were actually the ones that weren't going to do very well. Now, these are exactly the same data, but like the former example, we took out some of those patients to follow-up. We just assumed they were lost, and we did the same statistical analysis. Now the blue curve, like it looks like it did before, but the yellow curve, the experimental, appears to be doing better. But it's not because the drug's working better. It's just because of loss of follow-up. So the incorrect conclusion could easily be drawn here. At the end of this study, because of imprecise follow-up, we might incorrectly conclude that this new treatment helps survival, but it actually doesn't. And this new treatment might be expensive. This new treatment may have additional toxicities to it. So there's a real impact here with respect to drawing the correct conclusion. You need proper follow-up on both arms of this study. This is one of the amazing things that shows you no matter what your statistical design is, no matter what the review process is, if we don't get good follow-up data on these patients, we can't draw the correct conclusions. Next slide. So what we need are complete and timely submission of accurate data and thorough documentation of eligibility criteria of actually patients on the study. Next slide. And of course, we always need lots of things. We need complete description of all treatment received, whether to protocol therapy or not, because we have to interpret the results when we're done for both positive and negative studies of what the impact of that treatment was. And we need complete description of what's going on with the patient at every evaluation to avoid some of the things I just showed you. Next slide. So this is my one mathematical slide for the talk, and I know it's late in the session, but I couldn't resist it. It's the effect of not having good data or adherence on clinical trials protocol, what its impact would be on the sample size if there were no biases, if it was just a variance situation. So this is the formula. It says what a sample size would need to be is equal to the sample size the statistician thought it should be divided by 1 minus r squared. So that seems kind of mathematical. So let's take a look at what this means. So if I thought we were going to have complete adherence to the study on the first row, so non-adherence was 0%, so everybody adheres to what the protocol was, the sample size the statistician came up with, we need 100 patients to answer this question. Let's go down to the third row. Suppose 1 in 5 patients, 20%, aren't don't adhere, or the study's not adhered to for 20% of the patients. Then the sample size to detect the same effect is 156 patients. So what does that mean? That means that not getting the data or not adherence to one patient has an influence that's even beyond that patient. So 20% of the patients has an impact of 56 with respect to the sample size. It seems a little bit strange in that sense, but it just shows you the critical importance of actually having all that data come in on every patient or adhering to protocols. In a way, I always think of this as kind of like, if you miss it on one patient, it's not just the value of what that one patient provides to this study. Actually, it's impairing the value of another patient. It has a bigger impact than just in terms of the inferences of just that one patient. So in other words, if you need 56 patients to address what happened on 20 patients, that means it has a bigger impact than one alone. And so I just think this is sort of striking importance of what quality data means. Next slide. So high quality data are essential for good clinical trials. You've heard this before, and your efforts are essential for high quality data. Next slide. So, but it's not just essential at the end of the study. You can't just we can't all just take a break and worry about this at the end of the study. We're looking at the data all the time. So there's accrual monitoring by the statisticians and the study chairs. There's adverse event monitoring by the statisticians, the chairs, the data coordinators. There's AE monitoring. There's monthly reports. We're doing interim analyses. There's the data and safety monitoring committee. So it's not just in 3 years we need the data right. We need the data in a timely way. Next slide. One of the groups that's looking at the data, the critical group, of the SWOG Data and Safety Monitoring Committee, and they're evaluating interim results, endpoints, and safety days. They're trying to make a decision whether a study should stop now or carry on, whether more patients should be accrued or not. They're evaluating data requests from outside to use for purposes for designing new study. So we need high quality current data to make those critical recommendations through the Data and Safety Monitoring Committee. Next slide. So I went a little bit over, but I also want to say we need high quality data, and I added here timely data to emphasize the fact we need that. Back up the slide bit. We need timely data that are essential for our clinical trials, and your efforts are essential for high quality data. I want to thank everyone for being part of this course. I always learn a little bit when I see aspects of this and the complexity of what you're being asked to do, but it really is fundamental for us moving forward. Thank you very much.
Thank you, Mike. Appreciate your presentation. Apologies for the mishaps earlier that we went over time, but we'll go ahead and stop here, and we'll see you guys in the next hour.